Your body makes estradiol, a very specific molecule, 17-beta estradiol. Now look at what a lot of women are actually prescribed: conjugated equine estrogen. Read that again: equine, collected from the urine of pregnant horses. It contains estrogens your body has never made and has no receptor built for, or ethinyl estradiol, the one in most birth control. A chemist took the estradiol molecule and bolted an ethinyl group onto it so it would survive your liver. That single change is why it hits your clotting factors harder than the hormone you make yourself. I'm Dr. Richard Nquenti. I'm a compounding pharmacist, board certified in anti-aging medicine, and I've spent my career on hormone replacement. Here's what almost nobody explains to you. Your estrogen receptor doesn't care about the brand name on the box. It cares about shape. Change the shape, you change the signal, and you change the risk. So the real question isn't, should I take estrogen? It's, which molecule are you actually putting in your body? And does it match the one you were built for?
Your body makes estradiol, a very specific molecule, 17-beta estradiol. Now look at what a lot of women are actually prescribed. Conjugated equine estrogen. Read that again: equine. Collected from the urine of pregnant horses. It contains estrogens your body has never made and has no receptor built for. Or ethinyl estradiol, the one in most birth control. The chemist took the estradiol molecule and bolted an ethinyl group onto it so it would survive your liver. That single change is why it hits your clotting factors harder than the hormone you make yourself. I'm Dr. Richard Nquenti. I'm a compounding pharmacist, board certified in anti-aging medicine, and I've spent my career on hormone replacement. Here's what almost nobody explains to you. Your estrogen receptor doesn't care about the brand name on the box. It cares about shape. Change the shape, you change the signal, and you change the risk. So the real question isn't, should I take estrogen? It's which molecule are you actually putting in your body, and does it match the one you were built for? Over the next half hour, I'm going to walk you through 90 years of history that nobody handed you at your appointment. Where these drugs came from, who made them, why they were designed the way they were designed, and why one single fact about patent law shaped almost every hormone prescription written in the last 80 years. By the end, you're going to understand your own prescription better than most people who write them. Let's start before the beginning. Long before anyone knew what a hormone was, physicians had noticed something. Remove a woman's ovaries, and she changes. Everything changes. Her cycles, her bones, her sleep, her temperature regulation, her skin, her mood. Something was coming out of that organ, and when it stopped, the whole body knew. So in the late 1800s, medicine tried the crudest possible solution: give the substance back. In 1889, a 72-year-old French physiologist named Charles-Édouard Brown-Séquard stood before a scientific society in Paris and announced that he had been injecting himself with extracts made from animal testicles. He claimed he felt stronger, younger. He was mocked, and he was also, in a strange way, right about the principle. Glands make something. That something can be replaced. By the 1890s, pharmacies were selling dried, powdered cow ovaries for women with menopausal symptoms. Ground up ovary in a capsule. It barely worked, because the active hormone is present in tiny amounts, and much of it doesn't survive the gut. But the idea was now loose in the world. The problem was that nobody knew what they were actually looking for. That changes in 1923. Two researchers in St. Louis, Edgar Allen and Edward Doisy, develop a way to actually measure the ovarian hormone. They use a biological test in rodents that gives a reliable signal. Suddenly you can tell whether a preparation contains the substance or not. You can compare batches. You can hunt. For the first time, this becomes a real chemistry problem instead of a guessing game. And then, in 1927, two German scientists named Aschheim and Zondek make the discovery that shapes everything that comes after. They find that the urine of pregnant women is absolutely loaded with estrogen, far more concentrated than blood, far more available than ovarian tissue. Sit with that for a moment. That single observation is the reason estrogen therapy has a 100-year relationship with urine. Not because anyone thought urine was elegant, because pregnancy urine was the richest, cheapest, most renewable source of the hormone on the planet. Remember that. It's going to matter. Now the race is on. In 1929, working independently, Doisy in St. Louis and Adolf Butenandt in Germany both isolate the first pure estrogen, estrone. A year later, Guy Marion in London isolates estriol. And in 1935, Doisy isolates the big one, 17-beta estradiol, the primary estrogen of the human ovary, the molecule your body actually makes. I want you to understand what that work cost. To get pure estradiol, researchers processed literal tons of animal ovaries. Tons. To produce a quantity of pure hormone you could measure in milligrams. A speck. Butenandt would share the Nobel Prize in Chemistry in 1939 for his work on the sex hormones. So here is the first thing I need you to take away from this entire video. The molecule was never a mystery. We knew exactly what the human body makes, and we knew it in 1935. Before penicillin was widely available, before the polio vaccine, before the structure of DNA, we had the human estrogen molecule fully identified and characterized. The problem was never knowing what to give. The problem was supply. And supply is where the money enters the story. In the early 1930s, a brilliant biochemist named James Collip, working at McGill University in Canada, develops the first commercially viable orally active estrogen product. It's called Emmenin. It's derived from the placenta and urine of pregnant women. And it works. Women take it, and they feel dramatically better. There's just one problem, and it's fatal. Your entire manufacturing supply chain depends on collecting urine from pregnant women. You cannot scale that. You cannot industrialize that. The product is astonishingly expensive, and only wealthy women can access it. So the manufacturer goes looking for a bigger source. They need a mammal that produces enormous quantities of estrogen during pregnancy, that can be housed, managed, collected from every single day at industrial scale without asking anyone's permission. And they find her. A pregnant mare. Pregnant mare urine. Premarin. That's the name. It was never a secret. It was never hidden in fine print. It is printed on the box, and it has been for 80 years, and almost nobody has ever stopped to read it. In 1941 in Canada, and 1942 in the United States, that product comes to market, and it goes on to become one of the most prescribed medications in American history. For decades. At its peak, tens of millions of prescriptions a year. Now I want to be fair here, because this is exactly where a lot of online content becomes dishonest. That drug helped an enormous number of women. Hot flashes, night sweats, sleeplessness, bone loss, it worked. It is a real potent estrogen, and it does real things in the human body. Women who were suffering got relief. That matters, and I'm not going to pretend otherwise. But look at what's actually in the capsule. It is not one molecule. It is a mixture of roughly 10 or more different estrogen compounds. Estrogen sulfate is the largest single component, but a substantial fraction is equilin sulfate, and equilin is a horse estrogen. So is equilenin. So are several of the others. These molecules exist in a mare. They do not exist in you. Your body has no pathway that produces them and no established pathway optimized to clear them. And yet they bind your estrogen receptor. Some of them bind it tightly. Some of them persist in the body long after dosing. So please understand, this is not a story about natural versus unnatural. Horse estrogen is completely natural to a horse. The question is whether it is native to you. And it is not. Now the story takes a dark turn in the 1970s, and this part explains something you're going to need later. For decades, this estrogen was given to women largely by itself, unopposed. And in 1975, 2 studies published in the same issue of a major medical journal found the same thing. Women taking unopposed estrogen were developing endometrial cancer, cancer of the uterine lining, at substantially elevated rates. The reason is simple biology. Estrogen tells the uterine lining to grow. Progesterone tells it to stop, mature, and shed. Give a woman with a uterus estrogen alone, year after year, and you are giving a growth signal with no counter signal. Prescriptions collapsed almost overnight, and the industry's answer was to add a second hormone to oppose the estrogen. But, and this is the hinge of the entire story, they did not add progesterone. They added a synthetic progestin. Hold that thought, because to understand why, we have to talk about the single most important fact in this entire field. If this is already changing how you think about your prescription, take 1 second and hit like. It sounds small. It isn't. It's what pushes this in front of the next woman who's been told her options are the pill or nothing. And subscribe, because I go this deep every week. Now, here's the fact that explains everything. Estradiol was published in 1935. Once a molecule has been described in the scientific literature, it belongs to everyone. It is no longer new. It is no longer novel. Nobody can claim it as an invention, because nature invented it, and a journal already published it. Which means no company on earth can own human estradiol. Not in 1940, not in 1960, Not today. It is permanently, irrevocably in the public domain. Now put yourself in a pharmaceutical boardroom in 1940 and think it through honestly. You can spend years and a fortune developing and testing a hormone that any competitor can copy the week you launch it. You carry all the cost, you keep none of the advantage. Or, you can change the molecule, just slightly, one small chemical group in one position, and suddenly it is new, and new things can be patented, and patents can be defended for years. Now watch how fast this happens. 1938. In a laboratory in Berlin, two chemists named Inhofen and Hollweg attach a small chemical group, an ethinyl group, to carbon-17 of the estradiol molecule. And I want to be scrupulously honest with you here, because the honest version of this story is far more interesting than the conspiracy version. That modification solved a genuine problem. Swallow plain estradiol, and your liver destroys most of it before it ever reaches your bloodstream. It's called first-pass metabolism, and it made oral estradiol therapy impractical. The ethinyl group blocks that breakdown. The hormone survives the liver. Now it works as a pill. That is real pharmacology. That is a genuine scientific achievement, and the men who did it were solving a real clinical obstacle. But notice what else it is. It is novel, it is patentable, it is ownable. And ethinyl estradiol goes on to become one of the most widely consumed synthetic hormones in the history of the human species. Billions of doses. It is still, today, the estrogen in most combined oral contraceptives. Now here is the counterexample, and I include it deliberately, because it's the part that proves I'm telling you the truth rather than selling you a villain. That very same year, 1938, in London, a chemist named Charles Dodds synthesizes a completely artificial estrogen. It shares almost nothing structurally with human estradiol. It's called diethylstilbestrol, DES, and Dodds makes a decision that is, on its face, genuinely admirable. He refuses to patent it. He wants it cheap. He wants it available to everyone who needs it, not locked behind a monopoly. And that is precisely what happens. Dozens of manufacturers produce it. It costs almost nothing. It spreads everywhere. It is given for menopausal symptoms, for lactation suppression, for prostate cancer, and eventually, catastrophically, to pregnant women to prevent miscarriage. It was prescribed to millions of pregnancies. In 1971, researchers report a rare and aggressive vaginal cancer appearing in young women in their teens and 20s whose mothers had taken DES while pregnant with them. Not the women who took the drug, their daughters. So let me be very clear about the lesson here, because it is not the lesson the internet usually draws. The problem with DES was not that it was patented. It wasn't patented at all. The problem was that it was a molecule with no natural counterpart anywhere in human biology, given to millions of people before anyone understood what it actually did inside a developing body. The danger was never the patent. The danger was the unfamiliar molecule. Keep that distinction. It's the one that will protect you. Now the pattern accelerates. In the 1940s, an American chemist named Russell Marker works out how to produce steroid hormones cheaply from a wild yam growing in the mountains of Mexico. Overnight, the raw material for hormone manufacture goes from precious to abundant. The supply problem, the problem that had defined this entire field for 40 years, is solved. So ask yourself, with cheap, abundant steroid raw material finally available, what did the industry build? More of the identical human hormone? No. Modified hormones. In 1951, in Mexico City, a team at a company called Syntex synthesizes norethindrone, a progesterone molecule that has been chemically altered. It is patented. It becomes the backbone of the oral contraceptive pill. At almost the same time, a chemist at another company synthesizes a different altered progesterone, which becomes the first approved birth control pill in 1960. And in 1959, medroxyprogesterone acetate comes to market. Another altered progesterone, another patent. That is the molecule that got added to horse estrogen after the endometrial cancer scare of 1975. Not progesterone, a patented synthetic progestin. Meanwhile, the horse estrogen product enjoys something very close to a monopoly for decades. And here's the fascinating part. Not primarily because of a molecule patent. Because it's such a complex, incompletely characterized mixture that when a generic manufacturer tried to produce a copy in the 1990s, regulators concluded they couldn't even define what a generic equivalent would have to contain. The complexity itself became the moat. And human estradiol, the molecule nobody could own. The money went into the only part you could patent, the delivery system. The transdermal patch, first approved in 1986. The gel, the spray, the vaginal ring. That's the whole story in one sentence. If you can't own the molecule, own the packaging. Now let's talk about why this isn't just history. Because none of it would matter if the modified molecules behaved identically in the human body. They don't. Go back to that ethinyl group, the one added so the hormone could survive your liver. It works exactly as designed. But think carefully about what surviving your liver actually means. Your liver is where clotting factors are manufactured, where inflammatory proteins are made, where sex hormone-binding globulin is produced, where triglycerides are handled. An estrogen specifically engineered to resist hepatic breakdown sits in that liver longer and pushes harder on all of those systems than the estradiol your own ovary makes and your own liver clears normally. That is not my opinion. That is the mechanism the molecule was designed around. The whole point of the modification is hepatic persistence. The clotting signal isn't a mysterious side effect, it's downstream of the design. And this isn't only about synthetic versus native, it's also about route. Swallow any estrogen, even perfectly bioidentical estradiol, and the entire dose passes through your liver before it reaches the rest of your body. Deliver that same identical molecule through the skin, and it enters your bloodstream directly, bypassing that first pass almost entirely. Same molecule, different door, different physiology downstream. Large observational studies out of France have consistently found that transdermal estradiol carries a substantially lower clot signal than oral estrogen. The same body of research found a meaningful difference in breast cancer signal between micronized progesterone, the molecule your body actually makes, and the synthetic progestins. And I'm going to be completely straight with you about the strength of that evidence, because you deserve that. Those are observational studies, not randomized controlled trials. They are suggestive, they are biologically plausible, they are consistent with the mechanism, they are not proof. Any clinician who presents them to you as settled fact is overselling, and you should notice when someone does that, including me. Now let's talk about 2002, because that year did more damage to women's health than almost any other single event in modern medicine. The Women's Health Initiative was a large randomized trial, and one arm of it was stopped early. The press conference was dramatic. The headlines were worse. Hormone therapy causes breast cancer. Hormone therapy causes heart disease and stroke. Prescriptions collapsed. More than half of American women on hormone therapy stopped within a couple of years. An entire generation of physicians was trained to be afraid of these drugs. An entire generation of women went untreated, through symptoms, through bone loss, through fractures. So let's look at what that trial actually tested. It tested one combination, oral horse estrogen plus a synthetic progestin, one route, one molecule pairing, and critically, in a population whose average age at enrollment was over 60, many of them more than a decade past menopause. That is a very specific experiment. It was reported as a verdict on the entire concept of hormone replacement. Here's the part that rarely made the headlines. That same trial had a second arm, women who'd had a hysterectomy, and therefore took the estrogen alone, without the synthetic progestin. That arm did not show an increase in breast cancer. In long-term follow-up, it trended in the opposite direction. Think about what that implies. If estrogen itself were the carcinogenic driver, you would expect the estrogen-only arm to show the signal most clearly. It didn't. And when researchers went back and analyzed the data by age, a pattern emerged that has since been supported by other trials. Women who begin hormone therapy near the onset of menopause, under 60, or within about 10 years of their last period, have a substantially more favorable risk-benefit profile than women who start 15 or 20 years later. There's now direct trial evidence that estradiol given early behaves differently in the arteries than estradiol given late. Timing is not a footnote. Timing may be the whole thing. And in long-term follow-up published 15 years later, neither arm of that trial showed an increase in overall mortality. Let that sit. The trial that frightened millions of women off treatment did not, over 18 years, show that those women died sooner. We took a finding about one formulation, one route, and one population, and applied it to all hormones for all women for 20 years. That's not science, that's a headline. If any part of this has been useful, share it with one woman who needs it. Someone who was taken off hormones in 2002 and never told the story got more complicated. Someone being told to just push through. That share matters more than any algorithm. Now, let me clear up the biggest misconception in this entire field, and I want you to notice that I'm about to argue against my own commercial interest. Bioidentical does not mean compounded. Say that again to yourself. Bioidentical is a statement about molecular structure. Compounded is a statement about who mixed it. They are completely different claims, and the deliberate blurring of the two has made a lot of people a lot of money. There are FDA-approved, commercially manufactured, mass-produced products that contain the exact human molecule: estradiol patches, estradiol gels and sprays, vaginal estradiol, oral micronized progesterone, approved back in 1998, and since 2018, an approved capsule combining bioidentical estradiol and bioidentical progesterone together in one product. Those are bioidentical. They are regulated, they are dose standardized, batch tested, and covered by most insurance. For the large majority of women, that is exactly where this conversation should start. So what is compounding legitimately for? It's for the gaps: a dose strength that simply isn't manufactured, a patient with a documented allergy to a dye, a preservative, or a filler in the commercial product, a delivery form somebody genuinely can't tolerate. Those are real, and that's the work I do. What compounding is not, is automatically superior. There is no good evidence that a compounded bioidentical formulation outperforms an FDA-approved bioidentical one. In 2020, the National Academies reviewed this in detail and were pointed about how thin that evidence base is. I'd ask you to be skeptical of anyone who tells you the compounded version is inherently better. Ask them for the data, watch what happens, and I'll go further, because you should know where I stand. Be cautious with hormone pellets. They are widely marketed. They're extremely profitable, and once one is inserted, you cannot adjust the dose or remove it if levels run high, and levels frequently run well above physiologic range. The evidence supporting them is far weaker than the marketing suggests. One more honest note: you'll see compounded products marketed as bi-est or tri-est, containing estriol. Estriol is a real human estrogen. It's the one Marion isolated in 1930. But it isn't FDA approved in the United States. And the claim that these blends are safer or more effective than estradiol alone is not well supported. I'd rather tell you that than sell it to you. So here's what I actually want you to carry out of 90 years of this history. The molecule your body makes was identified in 1935. And from that moment forward, it could never be owned by anyone. Everything built afterward was shaped, at least in part, by that single fact. The ethinyl modification, the synthetic progestins, the horse estrogen with its protective complexity, the delivery system patents. Every one of those decisions happened inside an economic structure, where the identical human molecule was the one thing nobody could profit from exclusively. That does not make those drugs evil. Some of them changed the world. The contraceptive pill reshaped human society. Millions of women got real relief from real suffering. I'm not asking you to be angry. I'm asking you to understand that the menu you were offered was never assembled purely around the question of what fits your receptors best. Other forces were in the room, and now you know they were there. So when you sit down with your prescriber, you are equipped to ask 4 questions. Write them down. 1. Which molecule? Is this identical to what my body makes, or is it modified? And if it's modified, why is that the better choice for me specifically? 2. Which route? Is this going through my liver first, or through my skin? And given my personal clotting risk, my blood pressure, my history, which one makes sense? 3. Which progestogen? Is this micronized progesterone, or a synthetic progestin? Do I need one at all? Do I still have a uterus? 4. Why this one, for me, not for the average woman in a trial that enrolled women a decade past menopause, for my age, my symptoms, my history, my risk profile, my goals? You are allowed to ask those questions. You should ask those questions, and a good clinician will be glad you did, because the woman who understands her own therapy is the woman who stays on it correctly and safely. Because your receptors were never reading the label, they were only ever reading the shape. If this changed how you think about a prescription you're taking, or one you were scared off of 20 years ago, do 3 things for me. It's the single thing that decides whether this reaches the next woman searching at 2 in the morning. Share it. With your sister, your mother, your best friend, the woman at work who's been quietly exhausted for 3 years. Send it to one person by name, not a group, one person, and comment below. Tell me what you were prescribed, and whether anyone ever explained the difference to you. I read them, and the questions in those comments decide what I make next. Subscribe, because next week we go deeper, into progesterone, and why the difference between progesterone and a progestin may be the most consequential distinction in this entire field. Thank you for giving me your time. Go ask better questions.